Okay, so we are going to continue with Professor Tambet Teesalu. He is the head of the Cancer Biology Institute at the University of Tartu in Estland. And your title is already on the screen there, Precision Cancer Nanomedicine with Tumor Homing Peptides.
Okay. Good afternoon. It's really a pleasure to talk here on this famous meeting. It's first time for me, and I will definitely come back here. So yes, I'm based in University of Tartu, and to this audience, maybe it's interesting to know that that's where Carl Friedrich von Kupfer worked, so the founder of Kupfer cells. And yes, I spend mostly winters, also due to climatic reasons, in Sanford, Burnham, Prebys in La Jolla. And what I'm working on is development of homing peptides for tumor targeting, and not only. As you know, the concept behind systemic targeting is that of vascular zip codes. And this concept was coined by my postdoc supervisor, Professor Öki Roslahti, a famous Finnish cancer cell biologist, and my current collaborator. And the concept is that the vessels in each normal and diseased tissue express markers unique to that tissue, and that the luminally accessible part of this diversity can be targeted with specific imaging probes and drugs. And how we target this diversity, you can use, of course, various ligands, antibodies, aptamers, low molecular weight compounds, vitamins. But most people choose to use peptides for affinity targeting of nanoparticles. And peptides have distinct advantages for nanoparticle delivery. They are, of course, biocompatible, non-toxic. They can be synthesized in cheap manner and scaled up readily, and they typically bind to conserved binding pockets on target molecules and have bioactivity. And typical homing peptide has binding affinity in the low micromolar range, which is quite good for nanoparticle targeting. And we explore this vascular diversity by a technique that was first published more than 20 years ago. Of course, now we have many improvements in the technique, including application of next generation sequencing and use of long circulating phage libraries, but the essence stays the same. So we take peptide phage libraries. We like T7 phage because it looks more like clinically used nanoparticles compared to filamentous phage, which has very long rod-like structure. We inject this phage library of about 1 billion different peptides into live animal and take out the tissue that we are interested in, or tumor. And this gives us, of course, the phage that displays peptide with affinity towards the target. And really good thing about this approach is that it's completely in vivo. It's very agnostic. What you get from here has relevance. You don't need to think about whether it works or not in vivo. And it also helps you to find some really cool phenomena and peptides that you wouldn't come up with just by rational design. And one example is here. It's a peptide IRGV, now called as SEND1 and clinically developed. And this peptide is a cyclic peptide that homes first to integrins on angiogenic blood vessels in tumors, and then is processed proteolytically into this fragment that starts binding to neuropilin to trigger vascular permeability and penetrate into tumor tissue. And this peptide, I'm really happy to announce, has just entered clinical testing, and first patients have been dosed with IRGD. And as IRGD works with both conjugated and co-administered payloads, it's now in first approach used as a co-administered drug. So we take standard therapies, in our case for pancreatic cancer, so gemcitabine and Abraxane, and see what effect IRGD has on drug distribution and activity. And hope this will go well, because if it will, then it has relevance to targeting of many different types of nanoparticles that we know, have problems with targeting, also due to this heterogeneity in EPR effect and aspects discussed in this meeting. Now, I will just tell you and provide you some examples of ongoing work in my lab. The theme really is, in this vascular zip code business, that we are moving into more and more specific and precise targeting. So instead of talking about generic tumor targeting, we talk about tumor macrophage or matrix targeting, so it gets more and more fine, and that's really useful, I think. So first example, matrix targeting peptide that we call PL1, it's unpublished work. And this work was inspired by work in Tarja Neeri's lab on extracellular matrix targeting antibodies that are in clinical trials. And he showed in very nice series of studies that you can target extra domain B of fibronectin and tenascin C, various isoforms for tumor selective specific delivery. And then a PhD student in my lab, Prakash Lingasamy, asked the questions: what if we could develop small peptides that would do the same trick, and even better, if a single peptide could target both tenascin and EDB at the same time? And why target and why target have this kind of bispecific peptide? The reason is that would allow us to achieve more uniform tumor distribution of payloads. And also, there is a real issue with affinity targeting that we often don't think about, which is the receptor capacity. If you think, and this work has been done in Öki's lab by Sajid Hussain, he basically went and studied the quantity of available receptors in tumors. And it's actually in picomoles per gram tumor tissue, quite low. So you need all approaches that could increase this. So Prakash did in vitro biobanning on EDB and tenascin C, and then he was able to identify a peptide that came from this nicely enriched library that was binding to both EDB and tenascin C in specific manner, shown here by antibody inhibition of binding with one or the other antibody. And then we moved on and used our model nanoplatforms for studying the PL1 in vivo targeting, and we use iron oxide nanoworms developed in Mike Saylor's lab by Joe Park, and silver nanoparticles from our own lab. And first we found that systemic PL1 nanoworms home to glioma and other solid tumor lesions in mice in nice and very specific manner. Control nanoworms don't home. And again, that in vivo, if you use blocking antibodies, you can decrease binding using either of the antibodies and block by combination of two antibodies, quantified here. And then what we also want always is like a firm quantitative data on homing. And we have found this system really useful. So you have two types of silver nanoparticles which are isotopically barcoded. So silver 107, 109. One is with the peptide, the other is without, and then you inject them as a mix to an animal and then see what happens with the distribution of those particles, one or the other. And it's internally controlled and everything happens in the same animal, so you don't need to worry about inter-animal variability and those issues. And here is how PL1 performs. So this is homing of non-targeted nanoparticles to glioma, and this is PL1 targeted nanoparticles, same animal. This is probably due to EPR effect that we heard about, and this is specific targeting. And it's highly quantitative, very sensitive, and gives you firm numbers. And then finally, Prakash went on to show that the therapeutic nanoparticles targeted with PL1 have efficacy. And this is really proof of concept study. We used CLAC pro-apoptotic peptide, which is not a fantastic payload, but makes a point. So now we are developing these PL1 guided particles towards more clinical applications. We want to set up companion diagnostic and then as a research see what effect IRGV co-administration may have. Now, I also want to show you again the power of T7 phase display in exploring the vascular zip codes, and this is work by Kadri Tome in my lab. And she's interested in BBB targeting. And we are newcomers in this field. We have been mostly doing tumor work. So our approach was to scan the literature and take a number of peptides reported to home and penetrate BBB on T7 phase. And then just mix those phases in equimolar ratio, inject them into animals, take out the brain and see what happens. And then one peptide clearly outperformed the others, and the sequence is here. It's a published peptide, no follow-up in the literature. It was just lying there. And then we moved on and did in vivo alanine scan and then constrained library screen and came up with improved version of that peptide. And now the bottleneck in all this homing peptide business is a receptor identification. And now we are pushing towards setting up a promiscuous biotin ligase-based system for standardized receptor identification. And time is up. These are the people in my lab. We still have a very fruitful ongoing collaboration with EUCI collaborators, funders, and we are looking for postdocs at the moment. So if you or somebody you know might be interested. Thank you very much.
Thank you very much. Very nice pictures for people who may be interested to join you
Yeah.
as a postdoc. Any questions to the presentation? Probably easier if I can. You have to.
Have you investigated how the conjugation chemistry of the peptide to the nanoparticles affects the homing? Is the orientation of the peptide important? Is it important to have a linker between the nanoparticles and the peptide?
Yeah. We, haven't invested a lot of effort into, testing different linkers and so on. We routinely use aminohexanoic acid linker, And this works in our hands pretty good.
Anybody else? And otherwise, I think you have to save your questions for the next coffee break, whatever. So.